So, for example, in our spinal cord, we have these different vertebrae, and they look different from each other. But the cells that make up those vertebrae are really identical. And so it's really nearly identical. And so it's the location in which these things are in, which gives them some special features. And changing, zip codes, changing where you live and what the rules are, and whether you can build a 2-story house or 1-story house or what color you can paint, those rules can change very easily. And that's what evolution acts upon. Cell was not well understood. Genetics wasn't yet described. And so I would come at the end to this idea of the origin of ideas here, and quote a couple of physicists just to throw in a mix here. But I really want, as I say, take a very scientific approach to what we can now add to Darwin's ideas, and I think satisfy those people who are puzzled by some aspects of evolution. It won't satisfy the people who made a career out of denying it, but at least it might help other people think about some of the issues. Let's see. So I want to also bring in a word which will come back over and over again, called evolvability, and that may be already sort of anticipating some of the answers, but there's something about life that is capable of evolving. There are features of living things which may explain why life has evolved as fast and intricately as it has. And this comes to the question of the nature of variation in biology and what kind of variation is generated. This is as Darwin sort of thought, in all possible directions, or are there certain kinds of variation that that can happen? And we kind of know that as we see the evolution of an automobile or an airplane, it doesn't go in all possible directions. It sort of builds on what it has in certain directions. And life, as we look at it now, with the perspective of 211 years since Darwin's birth, we definitely have much more information about the nature of biological processes. So this is the amazing question we want to answer, is that how can evolution be so successful and so creative? And you can imagine that a lot of people for a long time have thought that this is something beyond science, beyond magic, and needs a more spiritual or other kind of explanation. But even if you're not tempted to go in that direction, it's truly quite amazing what life has done on this planet. Now, Darwin's big ideas, which I think are two really amazing ideas, and which he documented, carried through, and argued in his work, was that life on Earth comes from a common origin. And he sketched this in his notebook on the voyage of the Beagle as like a tree, which was just branching off, and the branches were branching off. And that was a very original thought. I don't even know what drove him to that thought at such an early time. The other big idea is that the evolution proceeds by a process of heritable change. So he was relying, without knowing anything about genetics, he was relying on the fact that inheritance played a big role here, and variation occurred, and then there was selection on that variation. And it's quite remarkable in a short period of time what kind of variation you can see. My favorite are these Hawaiian honeycreepers, which evolved in a very short time that the Hawaiian Islands had had been created. Unfortunately, several of these are already extinct, and it's just sort of sad about that as well. So the question I want to raise, and I think you could be skeptical certainly of it even at this point, is biology built in such a way as to allow meaningful change to occur readily? And the other question I want to raise is when we see conservation, when we see things that do not change, does that mean that variation has reached an impasse? It could really change if anything useful. Or does it mean that despite continual change, or does it mean despite continual change, selection fails to operate? I mean, is that really what's going on, or is there some other explanation for conservation? And we'll come back to this, of course, and I'm sure many of you are familiar with not so much the variation we see in nature, but when you look closely how similar we are to all sorts of other organisms in extreme detail. So why have those things not changed, and why have other things changed? And I think we can provide some answers to that question. Now, the big problem with Darwin's idea, which he dealt with himself as the best he could, was how does a complex structure emerge, like the eye, from small evolutionary steps, which he felt were taking place, if it only becomes functional when all the parts are assembled. Now, it's one thing for a machine to become functional when the parts are assembled, because we've thought in advance about how all these parts are going to be put together and made them as separate units and then put them together so that it would work. But in this world without a creator, how did all these changes take place and then suddenly become functional and exquisitely functional? And he argued why this could be the case, particularly with vision. But again, it's something which forces many people to throw up their hands and say only a brilliant creator like God could really generate something like this. And the famous story that Paley, the minister, wrote about was, this kind of homily about finding a brass watch on a trail and looking at it, and even if it were broken, remarking how everything seems to fit perfectly. And since he knows this could not have been created by accident, like stone, he argued that it must have had a creator. And then he went to argue that the creator of life must be even bigger creator, a more creative creator than the person who fashioned this brass watch. And this has been used in one form or another as an argument that evolution cannot explain the variety of life on Earth. But it is a problem to have to explain what is the mechanism by which this happens. And I think we should face up to that. Now, as science has moved beyond in whatever the 1869 to 150 years since the origin of species, we've learned a lot about how things are made down to the atomic level. And this actually provides a lot of understanding that couldn't have been provided at the time that Darwin came up with his big ideas. First of all, we now know that steps can be large, not small steps, incremental steps. A few genetic changes can significantly change the phenotype. The phenotype means the anatomy or the behavior or the physiology, all the aspects of the operating organism. Even big changes are often rarely lethal. And that's quite surprising because you make tiny changes in a machine, grind off one of the parts of the little pieces of a wheel that goes around in a watch, and then the whole thing stops. But even big changes can keep the organism alive. And then I'm going to argue that novelty has a bias towards useful change. And that sounds to a lot of evolutionary biologists at first as it violates some general principles that even Darwin argued, that everything was random and useful change. Why would you select for useful change? But the fact is that biology has been developing life over and over and over again, and those things have been things which have been useful. And so there's a sort of bias about what kind of change can take place. Not everything is possible. You can't change, a sewing machine into a jet engine. You can make little changes around that concept of the sewing machine. So if you have certain ways of making changes historically in biology, that biases the kind of changes you get. And I'll do some examples of that. Okay, so we need a name for this process of efficiently generating the right kind of novelty. And if we can efficiently generate the right kind of novelty, selection, which was Darwin's big idea, becomes much more plausible. Because people say, well, how could you generate randomly something so exquisite as the eye? And we say, well, it's not everything can be created. There's certain paths that have already been sort of worked out. And so evolvability is where I put it at the beginning, is the capacity of the organism to generate novelty, a lot of it, with very little genetic change, or minimal genetic change, particularly novelty that's non-lethal, because lethal novelty doesn't do any good. It doesn't get carried forward in the population. And novelty that's likely to be useful. And I would expect that all of you would be uncomfortable with this last part. And again, I want to explain why we think that it happens. Now, how could this evolvability idea work? And I want to give 4 mechanisms that come out of our more or less recent understanding, the last 30 years, let's say, in biology, that explain something about the efficiency and nature of generating novelty. So let me talk about regulatory change. And regulatory change, by regulatory change I mean not changing the fundamental processes, but changing them by changing the time in which they happen, or the amount of which they happen, or the circumstance in which something happens. It's of a gene being used, not the structure of the gene product. So we see changes slowly in gene products, but we see much more extensive changes in how they're used. Let me just give one little example of how you can just change, emphasize the amount of something to get something very different. So this is the wild mustard, brassica, and it's been cultivated for many, many years. And if you select for the growth of the stem, you get kohlrabi. If you just select the lateral buds, you get Brussels sprouts. If you select for the terminal buds, you get cabbage. If you select for the flowers, you get cauliflower. For the stems and flowers, you get broccoli, and for the leaves, you get kale. All of this is there in fairly quickly, and by human cultivation, just selecting the amount, the relative amount of these components, you end up with all these delicious vegetables. So it doesn't seem, when you look at all these vegetables and think of them starting from scratch generating these things, it's more remarkable and less obvious how that would happen. When you see it's just how it can be done over and over again by selection for a plant that seems to have more stem or more sprouts or more leaves. Now, I want to talk about something a little more interesting maybe than vegetables. The evolution, invention of flight, as in invertebrate animals. I mean, it's also been invented, obviously, in insects. But how did this happen? I mean, is this it's happened only 3 times. There are only 3 forms of modification that have given us flight. One in mammals, the bat, the birds, and the pterodactyl. And I'm very proud of this picture of a pterodactyl. It's very hard these days to go out there and snap a picture of a flying pterodactyl. So I hope you appreciate this. All right. So how do you get the evolution of a wing? And mainly, how do you get the shape and structure of the wing? Well, so here we have the skeletons of a pterodactyl, a bird, and a bat. And if you look at it, in the pterodactyl, it just extended the bone of the 4th digit. 4th digit stand up here. The birds, however, did it differently. They extended the forearm and the wrist bones. And bats, they just extended the digits and also the forearm. So it was really, again, the invention, when you look at it underneath, is not so remarkable. It's just this, it's really, as I said, this regulatory thing that is extending, regulating the amount of something, how long something grows, the timing of something, rather than the invention of something completely new. So this is the regulation of length, not the basic machinery making a bone or the invention of new bones. That was the case. Now, the big surprise at the end of the 20th century was how similar living things are. The human genome is only about 21,000 genes. That's only about 1.5 times the number for fruit flies and only 6 times the number for a small bacteria that sits in our gut. 15% of the human genes are shared with the bacterium, 25% with yeast, 50% with flies, and 70% with frogs. So on the level of gene products, there hasn't been so much invention. But how they're used is where the invention takes place. It shouldn't be, for people at a place like MIT, such a big surprise, because we have computers. Now they've improved, and that's all fine. But fundamentally, they're just simple switches, but yet the computer programs, the regulatory machinery, gives you all sorts of things that you from the same basic machinery. And so regulation plays a big role in the invention of development of computer science, but it also plays a big role in animals. Okay. So it's most change in evolution is the when, the where, and the how genes are used, and not the genes themselves. There are some changes in genes, of course.
Can I ask you a question?
Yes. Yeah. In fact, I'd be happy if people want to interrupt. Because we don't want to go too long, but
I just wonder, why are you assuming that that number is not a significant number? Maybe that difference in number of genes is actually pretty significant. I mean, it may not seem significant if you just say it's 1.5 times the number for fruit flies, but that number may actually not capture how significant.
Yes, no, I don't want to diminish the significance of that. I want to say that in the 2 things are going on. Genes are changing. There are new genes, and they're important. But a lot of things were accomplished in these early stages that get used further on. And a lot of things are like the various forms of wings, where they're not new bones. They're just really the length of growth. So yeah, I appreciate that. Okay, but there's more to making a limb and bones. We have to, let's not just appreciate that. We have to power the bones with muscles. They have to be in the right place. We have to control the genes with nerves. They have to be in the right place. We have to feed the limbs with a blood supply. How do all these things happen in the right way to allow the formation of a new structure like a wing? And here we come to, I think, a really very important principle, which I could talk for many hours about, which I call exploratory behavior. And it's really a very biological principle. I don't know that it exists, by a stretch you could say it exists in chemistry, but a real stretch. So let's talk about what is exploratory behavior. So the first thing was about regulatory change, time, amount, circumstance where something's used. Here we're talking about a process. It's a non-genetic process. It's happening in all of us. Of random variation followed by selection for a functional outcome. There are many futile paths are taken to maximize the chance of finding a functional one. And I think evolution itself, in Darwin's view, was a quintessential exploratory process. Lots of variation, select on the ones that work, retain those. But this kind of concept exists in many of the tissues and processes in life. So this is one of my favorite. It's about ant foraging. So you have to pity the poor ants. They're virtually blind. They can see light and dark. They can't image things. They're going to go out to find some luscious dead cockroach out there that they want to bring back pieces to the nest. And how do they do it? How do they generate what looks like a very intentional path? I mean, an ant trail that goes from the anthill to this cockroach, over hills, around little things, until they get to this thing. Did someone get up at the top of the anthill, look out with binoculars and said, march in this direction and you will find something worthwhile? Oh, I just told you they're virtually blind. And so the way they do it, and some ants at least, is they go out on missions. And then if they don't find anything, they turn around and go back to the anthill. And they find their way back the same way Hansel and Gretel did. They leave a trail behind them. In this case, it's a volatile odorant. So it doesn't last forever, but they can, they go out until they get frustrated, and then they turn back and they follow that trail back to the ant. This ant was not very successful. Next ant also failed to find anything. But the next ant finds this dead cockroach or insect, and when it goes back to the anthill, it reinforces the trail because it knows it found something useful. And ants tend to follow existing trails. So what started out as this random exploration ends up with a reinforced, looking like a very deliberate process. Okay, so this is random variation and selection occurring at this level. Now, in making a limb, all this other stuff, which is important and has to be in the right place, isn't being put in that place. It's finding its way in all of us when we developed it from embryos by a process of exploratory behavior. So, say the bone leads. Let's say a piece that's the bone on the lower right there. The muscles migrate from some tissue called somites, and the myoblasts migrate out, and if they find the bone, they stay there. If not, they keep migrating around until they find something, or they just die out. The nerves do the same thing, and they are the nerves that fail just die out. But the ones that succeed make synapses and function. And the blood supply also does the same thing. This is where VEGF plays a role in the whole process. So it's a little like ants. What's really going on is with nerves, let's say, is there's a target. The target is secreting something. And if it were a blood vessel, a region that isn't getting enough oxygen would be secreting something. And the nerves would be out migrating. And if they hit that, they stay there. But if they don't, they die away. And then you have something that looks like an intentional structure that has been generated, but it's really been generated by variation selection. And for those of you who know more biology, the adaptive immune system in our body is also, it looks like an antigen has come in here and somehow instructed the body to make an antibody against it, but instead it makes zillions of antibodies randomly, and the one that happens to interact with the antigen gets reinforced, and that's how it works. So it's not an intentional system. So this makes the development of new limbs and appendages much easier to achieve, because you don't have to change all these systems. You move a bone in another place, and they'll find their way. Okay, a third process which contributes to evolution is compartmentation. And here it is that, we only have, say, 22,000 genes, 21,000 genes. And how are we going to accomplish all the variation that we have in our body? And the way it happens is our body is actually divided up into domains, regions. And those genes get different instructions in those regions. So this gives us a lot. We can both vary the kinds of size of those regions. We can also vary, we also have many different cells that are going to inhabit those regions. So each domain behaves differently in development, and the rules in each compartment can change without changing the genes. And this makes it easier. So in a fruit fly, you can actually stain for these things. These cells all look the same, but they're different they're colored differently because of the dye that's being used, or the probe that's being used. But in fact, they all look the same, but they're different. Because they're in different regions, and they will do different things in those different regions. So in a fruit fly, you get these domains, each of which has this like a separate code, like a zip code. And if you found yourself in that zip code, one zip code, it'll behave one way, and in this other zip code, it'll behave another way. And you don't change this code, really at all. But you change the rules on which these cells will have. And you evolve those, change, another form of regulatory change. So, for example, in our spinal cord, we have these different vertebrae, and they look different from each other. But the cells that make up those vertebrae are really identical. And so it's really nearly identical. And so it's the location in which these things are in, which gives them some special features. And changing, zip codes, changing where you live and what the rules are, and whether you can build a two-story house or one-story house, or what color you can paint it. Those rules can change very easily, and that's what evolution acts upon. These compartments are like state political boundaries. They're arbitrary. They don't pay attention to geography or education or language in many cases. And each of these political divisions can have its own rules and evolve differently or take on different functions. And this is and in fact, because these compartments were established a long time ago in evolution, they don't make any sense any more than the map of Africa looks. Political map of Africa makes any sense. They cut across wings and half of parts of legs, and but having separate regions means you can deliberately instruct the genes what to do in those regions. All right, the final mechanism is I think, the most important, but the hardest really to explain. And we've called it weak linkage because it means that things are not so fixed in the way they interact. And so what is weak linkage? It's a feature that allows for easy evolution and new input-output relations. And so biology has built these linkages to be very, very easily removed. And I'll start out by giving you an example from our world. So let's just talk about the electricity grid. The electricity grid was, was developed well before we were born. And it comes with a plug, and you can plug things into this outlet. And you can plug things into this outlet that were never conceived of at the time when the electricity grid was created. And the more things will come along, and we'll still be plugging them into outlets. And the same is true, actually, of power generation. We change the ways we generate power. We don't have to change the electricity grid. We don't have to change the way in which we plug things in. So this kind of thing facilitates change. It makes it easier to change things. And biology has the same sorts of mechanisms. If we had to change the electricity connections every time we came up with a new electrical appliance, or it would really slow down things quite a bit. And there are many, many things in our industrial life that are like that. I mean, when people first made railroads, they made them with different gauges. They were better for going up mountains or going on flat land. And they then decided this is not worth it. If we can make them all the same gauge, we can move cars from one railroad to another railroad and facilitate those kinds of changes. The same thing is true with couplings between railroad cars, which are also different. And of course now become Wi-Fi. So what this says is that the connection between the action and what we act upon is very, very easy. And so it's like starting an engine. All you do is turn the key. The engine is very complicated. We don't understand the engine in a car. It changes all the time. But turning the key is something we can do very easily. And so these kind of processes in biology are like the key turning. They get a lot of things to happen in response. They don't have to be engineered for each new application. So this is a general principle that kind of emerges. In life, we're used to taking orders. And if we're used to getting signals that tell us exactly what to do. But the signals in biology are really simply permissive. They don't tell us what to do, they just allow us to do what we want to do. And that kind of permissivity is easily rearranged, and because there's no complicated ordering system that's going on. It's like the plugging into the socket. And so that's the secret of living things, is this permissive signals. Okay. Do they help explain the tempo of things? So I'm going to just go back to the limbs and appendages for a moment. So here we see this skeletal structure of a human, a cat, a whale, a bat. And let's assume that there was a lot of invention around making these bones the length they are and what they were. But of course, we need to not only have the skeleton, as I pointed out, we need to have blood vessels and muscles and nerves. And because these things are adaptable, that is, they don't require specific instructions, they take their input from the actual by randomly exploring the territory. It means that we can, that there isn't much of an obstacle to evolution involving new limbs and appendages. And I think that's only become clearer in the last, 30 or 40 years. Okay. So I want to just go back to Darwin's theory and modernize it a little bit, but he would recognize this as such. That organisms and populations vary genetically and consequently vary in their phenotype, their traits that affect their fitness. In competition with one another or with the environment, the most fit are the ones that contribute the most progeny to the next generation. And the process selects a better adapted subset that carries a different subset of genes and manifests a different set of properties. And then we say the population that has evolved under selection. That's the modern consensus view of what Darwin said. But if you look at this, there really were, I guess the question, what do we need to add to Darwin's theory? And I would say that there are really three theories here that are implied in what he's saying. We need a theory of heredity, we need a theory of selection, natural selection, and we also need a theory of how the phenotype is generated in the development of the organism. Now, Darwin didn't know about heredity, about Mendel's laws, and Mendel was working on the peas at the time Darwin was working on his Origin of Species, and nobody knew about that until 1900 when Darwin was dead, of course. And Darwin he knew he needed a theory of heredity. He came up with a terrible theory of heredity, and it was hailed at the time as Darwin's second greatest accomplishment, but it's now just an embarrassment to look at. So, but he knew he needed a theory, and we now have a good theory of heredity. His theory of natural selection has persisted as being quite sensible. He knew about breeding. There's lots of examples in nature of natural selection, so that's pretty well understood. But we need this theory of how the organism is generated, how easy it is to change, what sorts of structures can be made, and that coupled with these other two theories would then fulfill Darwin's ideas. And coming back to the skeptics, the biggest question for skeptics and even scientists is how likely is it that useful variation can arise on which to be selected upon. The selection is not a big surprise. When you have it's just like breeding is not. Once you have an animal that looks like, or a plant that looks like what you want, and you breed it and let it multiply, then you get that organism. But how do you get that variation in the first place? What I've tried to argue is that getting variation is not as difficult as it sounds, because a lot of things just are randomly exploring, and some things are adaptive and enable things to persist, and others of them just involve regulation, time, manner, place. Okay, so so I want to now talk about the last thing here, which is evolvability and conservation. That is, why are we so similar to bacteria and yeast and fruit flies? Why have so many things not changed? And there's the two ideas. One is that conservation implies constraint. Things don't change because it would be too difficult to change them. And that's, kind of embedded in this. We've got something, it's like our keyboard. We're also used to that keyboard. Changing it is very, very difficult. So when we see that, even though it's true the keyboard was developed for an odd reason, which was not to make typing more efficient, but to make things less efficient. Nevertheless, when we see that, and we tend to think that if something doesn't change, it's because it's hard to change it. But there's an alternative. And that is, conservation could instead imply deconstraint. It's the opposite. Things do not change because they're continually useful, continually selected to provide the functions that support change. And that was kind of the example I gave you with the electric plug, or the common rails gauge. And so many of these things don't change, not because they couldn't be changed, but because they actually continue to be useful in enabling change around them. You know, as I said, the electric plug is a good example. All the electric things we have nowadays that are plugged in, we're not even conceived of. So those are the things that I try to emphasize here today. This kind of evolvability, that is, functions that have proven useful over the millennia to support change, like electric plugs. And that's a much more interesting thing than just saying it's too difficult to change. And we know about constraint and deconstraint, and from our everyday life, it's really useful to have nuts and bolts that are the same gauge, and we don't have to. We can screw things together. If every device was made with a different nut and different bolt, it would be much more difficult to assemble things. Lego is a perfectly wonderful example. You can make parts simple in there, or you can make anything you want out of Lego without having to change the basic structure. And so I think there's a lot in biology which has this evolvability thing. And even the portable keyboard we have allows you to do so many things with it. It's pretty impressive. And so what is the history of life in regards to evolvability and constraint and deconstraint? It's a history where there is rampant invention and then stasis. So 3.8 billion years ago, life was invented on this planet, or somehow came to this planet. And this life had metabolism, DNA, RNA, protein, all of that was invented at that time. And now, it hasn't changed for over 3 billion years. This is the material that you get in a college biochemistry course. So maybe that's why people find it a little old and tiring. But about 2 billion years ago came the origin of eukaryotic cells, cells with nuclei, cells with mitosis, lots of features that made the cells of our body, which are also the cells of yeast and the cells of paramecium and cells of all these other things. They came up 2 billion years ago. They didn't change most of the metabolism and DNA of the stuff that happened, 3.8 billion years ago. But they also didn't change very much after that. That's why we can learn so much from this kind of stuff in cell biology course. And then finally, about 1 billion years ago, came multicellularity, and that involved all sorts of communications and signaling between cells. And again, that came up, and that didn't change. And then we have origin of limbs and body plans. We have something like 31 phyla. By the time the Cambrian, which was 530 million years ago, 30 of those 31 phyla were already invented. Maybe all 31, we just haven't seen it. And so, why no new phyla? Why, if it's possible to phyla so easily then, why no new phyla? And I think the reason is the phyla, I think it's a moving front of innovation. So while there probably was an organism somewhere out there, 400 million years ago, working on a better body plan. But the other organisms were working on mouth parts and appendages, and they probably ate this one for lunch. Because, that's not where the progress was focused on. So we see all of these steps that have taken place in evolution have really provided a basic framework for another step of evolution after that. and each of them has brought a certain evolvability that still made use of as we go forward. All right, so I was going to summarize this a little bit and just a few things. I thought MIT reminds me of physics a little bit because we don't have physics over at the medical school. We have biology over there. Anyway, I think there's a lot that's been added to the new age of biology, but it's really necessary to go beyond the chemistry and physics of that to other principles, and that often they emerge independently of the basic physics understanding. And so we can't just extend what we know, even if we know it better and better. We need to come up with a concept of Darwin, and that was the importance of Darwin's theory of evolution. And I think also, as people are developing this idea of how actually change takes place and the evolvability, that also is another concept which remains to be seen how explanatory it is over the next years. So I want to go back to a couple of physicists here, or one physicist of the mathematician. So Phil Anderson was a condensed matter physicist, still living actually. He's been quite old at this point, as far as I know. And he pointed out that we're not going to get these ideas necessarily to be building up from deep understanding of the microscopic principle. He said the reductionist hypothesis does not by any means imply a constructionist one. The ability to reduce everything to simple fundamental laws does not imply the ability to start from those laws and reconstruct the universe. And I think that's a tribute to Darwin, who really stood outside the science of his time and saw a bigger idea, a bigger picture. Stanislaus Ulam simply said, "Ask not what physics can do for biology, ask what biology can do for physics." And I put that in there. Just twist the knife a little bit. But I think that that's kind of where we are, and I just wanted to. This book was mentioned, but I took the name of the person I was acknowledging off that slide, excellent. But all these ideas have come through almost 35, 40-year collaboration, you could say, but intellectual collaboration and sometimes scientific collaboration with John Gerhardt, who's a professor at the University of California, Berkeley. And we both started with biochemistry and then moved into more developmental biology, and then started thinking about evolution from that perspective, which is somewhat different from the genetic perspective. And this book is, if you're really interested in more of this, is written for. It doesn't require a background in science. On the other hand, it's short, relatively short. So if you really want to know more, I would suggest you look at that book. Oh, thank you.
So does the concept of evolvability imply that there be a favoring in the evolutionary process itself of organisms that will have a kind of adaptability, evolvability, that they'll be able to go in multiple directions, or
I think it's more about facilitating change, but not necessarily what the end point of that change is. So, it means that you have all sorts of things that can change in the organism, but if it's about reducing the lethality of a lot of that change, and therefore increasing the scope of what you can select for. And so I think it's not about channeling in certain directions, but it inevitably channels in this direction. After all, we have, if we look at even today, if you look at all the vertebrates that exist today, they all have 5 digits. I mean, some of them may be fused, like horses, but they're 5 digits. Now, why is that? I mean, actually, the first animals that crawled up on land had variable numbers of digits. But somehow 5 worked out as a very flexible, easily regulatable process. And out of this, you get, bat wings and flippers of whales and the human hand. So it's obviously those kinds of inventions are the locus where change takes place. I mean, there's no reason why we couldn't have more digits, but somehow or other, it's not as easy to regulate, not as easy to change. We just haven't made use of that.
If I can follow up on that, does that not beg the question, when people are born with quote-unquote so-called deformities, is that possibly a reenactment, or whatever the right word might be, of the kind of processes you're describing? And it's just, because we're so used to just 5, we think it's a deformity, but in fact it may be just this same process happening again.
Yeah, I mean, deformity, first of all, there are people born with 6 fingers, and they, the fingers are obviously vascularized. There are nerves that go down there are muscles that go down there. They tend to be a duplication of the 5th finger in their shape, but they, I mean, I think that's an experiment. All these mutations are experiments, and maybe that, but somehow or other, those experiments, even though they existed early on, didn't seem to compete with the 5-digit thing. Some of that may be just embedment. It's hard to change what you've already got going, but some of it may be just that's just not a very effective place to innovate.
When students ask you what is life, what is the answer that you tend to give? Or do you just tell them that's too broad a question?
If somebody asks me what is life?
Yes.
I tell them it's hard to explain, but you know it when you see it. I mean, it's a question. It's a question, and part of the reason it's difficult is that we don't have living things around that below a certain complexity. I mean, you go down to bacteria, we see the things I said. They don't have a nucleus. They don't have they don't differentiate some of them into different types of cells. Most of them don't. There are a lot of things we don't have. They're not multicellular, but they have metabolism, and they have they can make proteins, and they can make lipids, and they've done it so well that we just adopted and built on that. That was not a productive way. Well, that's not quite fair. Yeasts reinvented metabolism. We did not reinvent metabolism. All animals are basically very similar in their metabolism. They just took a piece of what bacteria had and didn't change. They thought it was much more useful to start building limbs and appendages and brains and other things that probably less important. We kind of get a lot of pride in them. So we have to.
I want to ask about the evolvability of these power socket type solutions that you describe.
Evolvability of the what?
Of these power socket type solutions that you describe as very useful for allowing other evolutionary directions to sprout from there.
Yeah.
But it's not always obvious to me how these solutions themselves would be selected for, given that it is not clear what their immediate fitness advantage is.
And given that evolution does not really see into the future, does not select for potential, could you go into a bit more detail as to how these solutions themselves first get fixed in a population?
I don't want to imply that it's all necessarily the most fit. It could be an accident. I mean, in other words, for example, taking the railroad cars, the coupling on the railroad cars, I'm sure there were better and worse ones, but in the end they wanted the same one. That was more important. And people could have, they did work on making better ones. But making better ones had to compete with the fact that everyone was using the same one. So I think it may not have been the best possible of anything. It was just that in order to move beyond that to doing other things, you kind of adopted a certain standard. I think it would be wrong to assume that everything has been optimized against all other possible options.
So whenever you hear people talking about the possibility of life on other planets, you hear them talk as if it goes without saying that life is going to eventually be intelligent. Especially when physicists talk about that. That's what sounds like what they assume. So as a evolutionary biologist, what is your view on that?
Whether there are other planets,
Whether if there is life on other planets, how that it is almost certainly going to be intelligent.
Yeah. Well, that would be a big discovery. I'd have to say life on other planets. But the step from non-living, simple chemical processes to an actual life form is still staggering to me, and to many people. And there are definitely people who think that life arose somewhere else, and came here with some contaminated meteorite carried some spores of something, and they came here, and we found life that looked like us, on Mars, for example, like living things here, we would be thinking about that as a possibility. It doesn't seem likely that this is the only way to do it. We have 20 amino acids. That's a good number, but people already know that you can get rid of some of these codons and get things that can be sort of functional, maybe with not all 20 amino acids in simple organisms. So, if it looked very much like us, it would really, I think, increase the likelihood that we're all products of some contaminating event. And but if it looked different, then we would be really a lot to think about how life originated. That would be quite interesting. Either way, it would be interesting, but it's yeah.
Well, let's think more about the evolvability of intelligence.
Evolvability of intelligence. Sometimes I wonder where we are. But the brain, is clearly operating not by new genes, but and not even by new cell types, but by connection, by evolving the connectivity. That's the main driving force, and the size of the organ. People speculated that really the early brains or the primitive brains were really not so much about looking outside, but it's just keeping track of what the rest of the organism was doing, how it felt, it was hungry, or it needed something. I mean, there was an internal, a big internal nervous system which is feeding into all of our tissues and telling us to communicate. So, from these simple ideas of some inputs, light, and using that, maybe vibration, mechanical input, sound, into internal monitoring, to what we call intelligence, are some very big steps that I don't think I have much to say on the subject, but it's a good problem for you to work on. Because I think
How do your ideas on evolution apply to the development of a fetus, for example? You have compartmentation, you've got exploration, like in the analogy of the ants, and in a certain sense, you could say that a single cell evolves to become an infant. How does that work?
Well, a lot of the things I talked about today were about multicellular development. And I talked about limbs and appendage, those things were really about how they get fashioned in evolution. And I didn't talk much about the brain or the heart or some of those things, but they're all, they're definitely processes. As far as how the fetus starts from a single cell and generates, I want to specifically say it was a good point, generates the animal. It generates a structure which has all these compartments set aside. Just like a fruit fly, only it has more compartments. Fruit fly has about 100 compartments, and a human has about 200.
Which were predetermined by the genome, right?
Predetermined by the genome and the whole, and those compartments are a kind of a rough map of where things are going to be and what should be happening in those places. But then beyond those, within those compartments, there's more and more cell interaction and parsing out different territories and differentiation and cell movement. I mean, it's like that map of Africa. It gives us the general lay of the land, but and tells us what countries are different. But within the countries, there are more subdivisions and more things that are happening, and that's the way it is in human development or any development. And the genes that are laying out the territories in the fruit fly, many of them are used in humans as well, and that's been conserved all the way up.
I was just wondering, you and probably most people when approaching the topic of evolution are analyzing from the angle of natural forces playing out, but I was wondering if you ever considered unnatural forces of economic and political organization and how that is influenced, shaped either enabling certain subpopulations to survive and thrive while disabling others, or how that plays into the selectivity process.
So you're really thinking about the effect on the organism of the environment and the social and political. But there's another. I thought you were going to go in the other direction, which was people have for many years tried to use life as a model for social organization, and that led to this period of social Darwinism, where
analogous, you can look at different scales and learn from
that sort of thing. And we could talk about that. But you're talking about how I just would say that the social structure is part of the phenotype. That we're and it's clearest maybe in something like an ant or a termite colony. Where you have all these different these individuals all doing different jobs, all to support the queen, support the growth of the colony, So lots of things are really, not only are they have they evolved this way, but they're maintained this way in the life of the colony. I mean, where the number of nurses and soldiers and things, that's really biological change. Because these ants and these are being affected by the environment, affected by signals from the food they eat, and the things that the queen may generate to these groups of animals, and so they change their phenotype depending on that. So that's, you could say almost, it's like people said this. Maybe it's a little misleading, but a colony is like a superorganism.
Organism can be likened to a nation, or a company. Actually, a corporation with only one single product would be like what an organism.
But I think in humans, it's
so that's why if you have a cell that's differentiated and becomes a bone marrow cell, but then because of the environment changing and augments.
But I don't know if you use this as a metaphor.
Depending on epigenetics, just a lot of experience.
Oh, I see. You mean actually affecting it.
But
well, I think stress is one of the things that does affect the population, and it has some, but it's not a genetic effect. It may affect the way the phenotype develops. But yeah, I don't. That is an effect, but it's not something I've been thinking about at all. Yes.
Has evolution come to an end in humans?
Pardon?
Has evolution come to an end in humans?
Why would you think that? I think I can imagine two Tyrannosaurus rexes sitting on the savannah saying, "Do you think evolution has come to an end? What could be more powerful? What could be?" No, I think. Well, it's there's a good question about whether we can control everything and change the course of evolution. We certainly are very good at destroying things, so we'll have a big effect. But I mean, humans are evolving as well. So I think we're going to.
Does that mean destruction is functional?
Pardon?
Does that mean destruction is functional?
Destruction.
Destruction is functional, according to life.
Construction.
Destruction.
Destruction is functional. Well, certainly a major force to generate, challenges. We're going to have, I don't know what kind of challenges we're going to face. Whether other organisms are in millions of years going to come up and take our place, that's very hard to know. But we're subject to the same pressures that underlying. Let's see. So we're going to go on to this. I'm happy to do it, but I just don't want to. Maybe a birthday cake.
So, a question on, a lot of people often talk about emergent properties as something that, for instance, if you have hydrogen and oxygen, you couldn't understand water, let's say the properties of water, based solely on the principles on the lower levels, hydrogen and oxygen. I was just wondering, since you gave that quote from biology and physics, where the reductionism does not imply constructionism. Well, it doesn't imply actual ability to construct. The question whether, if you knew everything on the lower level, you believe that in principle it would imply constructionism. That is, the deeper sciences, physics, not biology. That's if you understood everything on the lowest level, then the principles that appear to you as principles in biology wouldn't really be, they would be shorthand, so to speak, simplifications, and shorthands for the human mind, based on the true principles, which would be the lower physics principles. Or would you have a different way of looking at it? That's a good question.
If you've understood everything and you've been able to derive all the higher functions, then that would be a resistance proof, say. And I think Phil Anderson was saying, in a system which is more deeply understood, better understood than anything in physics, they haven't done it. They can't predict chemistry. that was his statement. I think if that's true for physics chemistry, I think we have a lot of stuff she wrote from physics and biology. And maybe the argument was not to discourage people from trying to do that, I don't really want to do that, but more to say that there is a lot to understand. And sometimes, more general analogical theories, speed things up a lot. And the industry's well enough to know whether perfect gas law is well understood in a chronological sense. There's people who study genetic cases, acoustic, mechanics, they have even thought to try to work out a model based on more physical process. They didn't have these general rules. So I think for all of us here, the coexistence with people, fundamentals, thinking very broadly about general theories like Darwin. That's probably very constructive. And there's a couple of and then you want to talk about you want to
Yes, Mark, thank you so much for your presentation, and I want to thank you for taking my question. I'm really excited and I'm optimistic about the future, and I'm really curious if we're not done evolving the human mind, and particular kind of like more, I'm hopeful for more evolution in higher functions, and that's what I want to ask you. I really believe that in the very near future, when there's that big wave of change in the workforce with automation, supply chain, and all that kicks in, and people are going to shift from doing jobs that machines can take over, and it's going to free the human to do more meaningful work. I think that is going to be a way to encourage human beings to step away from the lower functions, territoriality, fighting, discriminating against people, and focus on higher functions of the brain, logical, abstract thought. Now that you're doing more meaningful work and universal basic income, my question to you is it possible that we can really evolve as a species where we're utilizing more of the functions of the higher brain instead of the lower brain?
well, before we could evolve that way, we probably could do a lot. Argue it a lot. Play around with drugs and short circuits. I mean, both negatively and positively are affecting our phenotype. So I don't know the answer to that, but I do appreciate your optimism.
So, well, first of all, I thought it was interesting that you picked Stan Ulof as your physicist. For people who may not know, he partnered with Edward Teller and developed the Teller-Ulof device, which is better known as the hydrogen bomb. I know, but that doesn't bode well for the survival of the species. But nevertheless, I wonder, when we talk about the conservative piece to this story, could it be, and you had an interesting theory, I thought, about that, and its relation to being able to continue to provide for the evolvability dimension. I understood it correctly, but could it be that it's conservative because there's just no need for it, for that particular set of components, if you will, to evolve further because they're just fine just the way they are? And then the other question I had is, and I think it's sort of related to something someone else asked about the fetus. There's a notion that people who believe in God are particularly concerned about life in the womb and the fetus, and that you're taking a life if you have an abortion. But interestingly, or paradoxically, the ideas of evolutionary and evolvability ideas that you're presenting could actually make a case for life beginning like it's a continuum, so it's already underway, and so could that bolster the argument against, or the argument for life, setting aside when life begins, life is happening, and in a sense, it's not just people who believe in God who are concerned about that, it's people who might reject notions of God but still see life as beginning, or as in a continuum that isn't easily separated out in the evolution of the fetus, or the evolving of the fetus.
There's the ethical issues around abortion and all those things that come from issues that make humans special and have associated human beings with God. So the biological sense of this whole thing, I don't know if it speaks to that at all, but I think that it is sometimes worthwhile to think of the process in not just one generation, but a whole evolutionary process. And so we're all changing over time, but I don't know, I can't really speak to what you have to say, but I think it's good to think about these things from a somewhat detached point of view and not get caught up in the specific question of when life begins, because life is this is just a step. It's not that it is a discrete step, but it's partly connected to the evolution of the mother and father and their genetic variations. It's going on and on and on and on. So I don't, unless you believe in something particularly explicitly religious, you have to add something from the outside to make that distinction. Arbitrarily, I believe that birth, I believe that is the point where we're going to make a decision. It's certainly not. It's not any different from all the steps that are going on. Like you said. I think you want to look at the last question.
Right, you kind of mentioned the limitation of biochemistry.
Pardon?
You kind of mentioned the limitation of biochemistry explaining the evolution. So what would you think we should look at on the fundamental changes on the genetics?
I wasn't, I don't think I would consider myself still a card-carrying biochemist. So I just would say that a lot of what we teach in biochemistry was accomplished 2.8 billion years ago. And that was with all these metabolic and synthetic processes. And the big step with eukaryotes was definitely to make the cells a lot bigger, to make them much more able to communicate in sophisticated ways with the environment, and then multicellularity was another step. So I think these were really big. What's interesting is how little evolutionary change really happened once it reached a certain point. I mean, you can point to genes changing, but we didn't evolve a new metabolism. We didn't evolve a new way of making proteins. We said, "Oh, this is working. If we're better off working on something else." And maybe that's what's happening in agriculture as well. We've kind of accomplished maybe how to feed people and how to clothe people and how to take care of them. We should be working on something else. But I'll leave it at
Thank
that.
you.